So today we're going to talk about enzymes. So remember, enzymes are made of proteins. So that's why we're talking about this unit, and that their shape relates to their function. So the big thing that enzymes do is they are catalysts. So they speed up chemical reactions. So they're not actually part of the chemical reaction itself because they're not being used up or changed, but a catalyst helps speed it up. An example of this is there's a catalytic converter in cars. It's actually platinum, and it's catalyzing the reaction, turning carbon monoxide to carbon dioxide. This is just an example of a catalyst. It's with something there that helps speed up the reaction. So how they work is that there is an active site that is on the enzyme, and that recognizes specific substrates. And substrates are those molecules that interact with the enzyme in the active site. They're the reactants in the reaction. So the reactants interact with the active site on the enzyme, and then there is a product that's produced. Here is also showing our active site and then the substrate that interacts with the enzyme. Okay, so all reactions require a starting energy called activation energy, and what enzymes do is they actually lower that activation energy required to take place for a reaction to happen. So here we have the activation energy where the frogs have to jump over this really tall stack of rocks, and so it takes a lot of energy for them to get to the product. But with our enzymes, we have a smaller, lower activation energy required. Here's again, we have our reactants and our products, and then with an enzyme, it's going to lower that activation energy. Okay, so a big thing with enzymes is there's substrate specificity. So that means the substrate is specific for our enzymes. So the enzyme binds to a substrate, and it's not just any substrate, it is particular one based on the enzyme. So here we have like a specific shape. That substrate can fit into that active site shape and form what we call an enzyme substrate complex, basically where the enzyme and substrate have come into contact and are bound together temporarily. And then there's some sort of change, and we have our products, and those products are released. But it's that active site region where the substrate binds, and it's going to speed up chemical reaction, which creates the products, usually because it's helping to orient those substrates in the correct way for the reaction to happen. So the shape determines the function. So sometimes an enzyme needs a little help getting into the right shape, and so we have things that help enzymes get the correct shape. Cofactors and coenzymes, we don't need to memorize these examples or anything, but just know that there are things that can help enzymes. So they're working with the enzyme, and they can be non-protein things such as zinc or iron or copper. These are just metal elements. Or they can be coenzymes themselves, where it's another enzyme or another protein that's going to help make the correct shape. Okay, so here we have, this is showing more of what a protein actually looks like. And the active site here, there's binding sites in blue and the catalytic site in red. So these are going to help be where the catalyzed reaction takes place, but there's also binding sites to help orient that those reactants. Okay, properties of an active site that can affect binding, the physical shape and size, as well as chemical charges. So here is showing the shape of the active site, but the same idea with charges. We can have compatible charges, so we will have opposite charges from the active site and the substrate itself. A big thing that is helpful is looking at the names of enzymes. So usually we will have the substrate that it's interacting with be identified in the name of the enzyme itself. Also, enzyme names usually end in ACE, A-S-E. So the enzyme sucrase breaks down sucrose. Lactase breaks down lactose. So you can see that the substrate lactose is in here, the name, as well as ACE, telling us it's an enzyme. So what do you think glycogen synthase does? It synthesizes glycogen. So we have names of enzymes that can give us information about the function of that enzyme. Okay, enzymes, again, since they're not used up in that chemical reaction, they're still there. They're the same enzyme they started, or yeah, the same enzyme we started with then as we end with, so they're reusable. So like here on this conveyor belt, we can reuse that same enzyme, or here this person working over and over again. And again, the structure relates to the function. We can think about the structure of this spoon relates to the function of eating the soup. Well, if we change that structure, just like if we change the structure of an enzyme, it's going to impact the function of that enzyme. So ways we can change the structure and function of an enzyme is called denaturation. Okay, so this is when we have the shape being deformed, or we lose that shape. So the normal shape of the protein is called the conformational shape. And then we can have denaturation happen to where we're changing that shape of the protein. It's usually irreversible. So here we have a normal protein. It is folded in lots of different ways, and then it's denatured and it's all loose and we have lost that folding. This is what happens when we cook an egg. That white is actually denatured protein. That's why we can't reverse it. So we are denaturing that protein. So things that can denature proteins, temperature is a big one. So as the temperature increases, at first it starts to speed up the reaction because we have more interactions happening between enzyme and substrate. But then at some point we reach where the enzyme itself starts to fall apart and denature, and then we're going to decrease the rate of our reaction. So at first it increases just because temperature, but at some point we denature the enzyme and then it's going to fall off. We will have an optimal temperature range where the enzyme works the best, and this will depend on the type of enzyme as well. Different enzymes will have different optimal temperatures. And yeah, at extreme high temperatures, we have denaturation happen. Extreme low temperatures, we will still have the reaction happen. It'll just happen slower because everything is moving slower. So here's another graph to show what would happen. pH is another thing that affects our enzymes. So remember, pH is the concentration of hydrogen ions, and it's that logarithmic scale. So we can also have an optimal pH, but unlike temperature where we're just slow at the beginning, pH we'll have no function at the beginning because it will denature with a low pH and low denatured protein at high into the range. So extreme high and extreme low will have a denatured protein. And that's because those hydrogen bonds are going to be disrupted because of those hydrogen ions in the solution. So we want ideal pH levels. Otherwise, those enzymes will be denatured. And again, depending on the enzyme, we'll have a different optimal pH. Substrate concentration can also affect this. So initial increase in substrate concentration increases the reaction. We can think about we'll have more collisions happening the more substrate we add. But we can get to what is called substrate saturation. And then we'll have a constant rate just because there is not enough enzymes to make the reaction go any faster because we have reached substrate saturation. Product concentration can also affect the rate of the reaction. So increase in product decreases the reaction rate because matter takes up space and it gets in the way, and we have a lower chance of those enzyme substrate collisions. So here again, we can think about that product being produced is this purple stars. So the substrate is the red, and we have these 5 enzymes here. But over time, we get more and more and more products building up as the reaction continues. And at some point, it gets to where we're not able to have enzymes and substrate collisions happen very frequently. enzyme concentration. So if we have less enzyme, we'll have a slower reaction because there's just less enzyme around, fewer collisions. So here we're going to have less reactions happening just because there's fewer enzymes. And then as we increase, we'll have more reactions happening. But again, we will have a point where it's going to level off if our substrate is limited, because we'll run out of substrate to react with. There are also inhibitors that can affect the reactions. So competitive inhibitors, these are molecules that can bind to the active site of the enzyme. They can be reversible or irreversible. And if they're reversible, then the enzyme can function normally after the inhibitor leaves. And then irreversible, the enzyme function is lost completely. And increasing the substrate can prevent this. So here's an example of a competitive inhibitor. So here's the active site, and notice that this green competitive inhibitor is binding to the active site so that our substrate cannot. We can also have non-competitive inhibitors. They work similarly, but instead of binding to the active site, they bind somewhere else called the allosteric site. Okay, so here's an example of our enzyme. A non-competitive inhibitor binds to some other site called our allosteric site. That binding is going to cause a change in the shape of the enzyme, called a change in the conformational shape. Now that active site is no longer able to be accessible, and so it makes it so that this enzyme is non-functional and the substrate cannot be accepted. And then again, it's going to decrease the reaction rate. In this case, increasing the substrate won't prevent it because we now have a non-functional enzyme. So adding more substrate won't affect anything. Here's an example. So here we have our active site and the substrate, but the allosteric site, when it's bound by a non-competitive inhibitor, like here, the active site is unable to be accessed. So that's an example of a non-competitive inhibitor. Allosteric regulation, again, allosteric is a site other than the active site. So proteins function at one site is affected by the binding of a regulatory molecule on the other site. So we can also have, not only can we have inhibition happening, we can also have activation happening at an allosteric site. So sometimes those molecules that bind to the allosteric site activate, and sometimes they inhibit the enzyme. Just depends on the example. So we have cooperativity happening where one substrate triggers the shape change in another active site. So we can have that happening as well. Just some examples. Okay, but this is kind of our overview of enzymes and the different things we need to know about what are enzymes and how they can be regulated and the different things associated with it.